This is video 13 of chapter 10, part 2. So we have been talking about the different types of smooth muscle. Now we're going to look at the structure. So some of this is a little bit of review. So the cells are smaller than skeletal muscle. They are spindle shaped instead of being like a thread. And they have a single central nucleus, unlike skeletal muscle, which was multinucleated on the outside section. The filaments are arranged a little bit different. So with that, actin and myosin are present, but not in sarcomeres. So we do not have sarcomeres present. That gives us our non-striated appearance. So basically, they are in loose bundles in the cells. So they're spread out. And actin is attached to dense bodies in the sarcoplasm and dense areas in the sarcolemma, which is the plasma membrane. The dense bodies and areas are then attached to something called intermediate fibers or filaments in the cytoskeleton. So it kind of makes like an interconnected network of actin and myosin attached to the cytoskeleton. So if contraction happens, it kind of pulls in all directions and causes that whole sheet of muscle or bundle of muscle to squeeze together. So the arrangement of the filaments does not direct motion in one direction like it did with skeletal muscle. It more contracts the whole thing together, and if it is in a tube shape, it squeezes it all together. So it's not as precise in USM as it could be in MSM, which might be a smaller bundle or small sheet or single cells. Other ways that smooth muscle is different, we have no T-tubules and a lot less sarcoplasmic reticulum, which decreases our Ca+2 storage potential. There's no troponin on actin. And we have an increase in calcium channels and pumps. So if we look at how the process of contraction differs, we have a few slight changes. So because we don't have troponin, calcium binds to a protein called calmodulin. And then we start a series of reactions. Calmodulin activates an enzyme, myosin kinase. So myosin kinase transfers a phosphate. So it moves a phosphate, kinetic energy, from ATP to the myosin head. So its name tells us what it does. And then myosin ATPase, which we saw in the cross-bridge cycle, uses the phosphate for cross-bridge formation. So we end up with the whole cross-bridge cycle again. It's very similar with actin and myosin, but the process of getting it to contract is a little bit different. So contraction stops when another enzyme called myosin phosphatase removes phosphate from the myosin head. So we do not need to rely on as much calcium because we have calcium binding to this protein and it just starts a chain reaction. And we still have the same cross-bridge cycle with contraction, but the processes in which it is initiated and stopped is a little different.